Hi everyone. My name is Pedro, and today I'm gonna talk about Trypanosoma cruzi infection and some inhibitors that our group have been working to fight against this kind of disease. So the project was developed at USP, University of São Paulo. USP is one of the most important public universities of Brazil and Latin America. And the campus that I have worked and performed this analysis is located in countryside of the state in the city called São Carlos, as you can see, and the lab it's called NACMed. It's the group of medicinal chemistry of São Carlos under supervision of Professor Andrei Leitão and Carlos Montanari. Our group work with this cycle, design, synthesis, and test. And this cycle allows us to predict some properties of interest and optimize the compound synthesized. So we perform several analyses as cellular evaluation, physical chemical assays, for example, calorimetric assays, and many others. And my area of study is analytical chemistry. So this kind of analysis could be performed between all steps of this cycle. It's a very useful analysis. And specifically, my work is validation, development, and validation chromatographic method for the compound synthesites. And this validation is in accord with the official governmental guidance, and it's pretty close to the other guides as FDA and European Medicine Agency, for instance. About the Chagas disease, it's a tropical neglected disease, was firstly reported and described by the Brazilian physician Carlos Chagas, and is caused by the protozoa Trypanosoma cruzi, this guy here, and transmitted majoritarily by the bite of the insect Triatoma infestans, this guy here. In Brazil, is commonly called of barbeiro. And according to the DNDi, it's a non-governmental agency. It's and one of the most important agents to study and fight against neglected disease. Chagas disease is endemic of Latin America. It's present around 21 countries, and but has been infected around 6 million of people around the world, and 70 million of people are in the areas of contamination risk because in addition to the beetle sting being the main form of contamination, there are others such as ingestion of contaminated food, blood transfusion, and transmission from the mother to the newborn baby. This actually is the most common form of transmission in areas non-endemic or where the vector is controlled. And the life cycle of protozoa, we have three different morphologies. The amastigote here, it's infections and non-replicant uh, form. The trypomastigotes is infections and non-replicant when we talk about the beetle, the bug. And amastigote form, this form is replicant and infections when we talk about the mammalian host cell. So what happened is the amastigote form replicates, change to the trypomastigotes, and this form disrupt the host cell. So it can infect a new cell or a new insect vector. Then they change again to the amastigote form. So amastigote form is exclusively intracellular. And when we talk about the symptoms, actually we have three phases. Né? The acute phase, the first one, where we have no symptoms in the most of cases. Usually, when the symptoms appears, the patient may have skin inflammations, fever, head pain, breath issues, and among others. And it's possible that symptoms during between 2 and 4 months. The next one is indeterminate chronic phase. Here we have absolutely absence of symptoms, but the patient is be able to transmit the disease. So this phase may be during decades. It's possible that the patients live with this disease for many years, besides the no symptoms. They shows no symptoms. And the last one, the chronic phase, 30 or 40% of the infected people can progress to the final step, this final stage. And here, the patient may present heart issues that often leads to sudden death or progressively cardiac insufficiency. But in other case, the patient may present gastrointestinal issues and among others. So the treatment, it's a problem because less of 10% of infected people in the Americas are diagnosed, especially because the in the phases of this disease, we have no symptoms, né? In many cases, and less of 1% of these patients receive the antiparasitary treatment. We have two drugs available against the Chagas disease, benzonidazole and nifurtimox. And these medicines are effective only in the acute phase and sometimes in the beginning of chronic phase. And these two medicines present several side effects. Several side effects. This scenario is worsened because infurtimox is banned in many countries, uh, especially because of these adverse effects on the neurological system. And benzodazole is commonly used, okay, despite of these side effects and the variable effectiveness among different parasite strains and this disease phase. As I said, it's effective only in the acute phase and beginning of the chronic phase. So because of this context, we need to find out new clinical therapeutic agents to fight against this disease. And neglected illness promotes health problems in developed countries as Latin American countries and Africa countries, especially in those with the economic and basic sanitation issues. Né? So, in this context, sustained purchase has been considered a macromolecular target for discovering new substance with antitrypanosome activity. And cruzipain is the cysteine protease with highest expression level by the trypanosome cruzi protozoa. So, cruzipain is essential to the survival of parasite and transmission of the disease, especially because this kind of enzyme is associated to the change of morphology of the parasite. So here we have a crystallography extrusory of cruzipain and one inhibitor, the ACE inhibitor. It's synthesized by our group. It's called NEC 0490. And this compound is a prototype to many other inhibitors that our group have been working. And it possesses a nitrile as a warhead and two amide bonds. So we can see some key positions, P1, P2, and P3 position that will interact with the subsites S1, 2, and 3, and the active site of the enzyme. And dipeptidyl nitrile, as I show in the last slide, has been recognized as efficient inhibitors of cysteine protease. This kind of compound has a moderate reactivity because form a reversible covalent bond with the activity site of the enzyme and has adequate toxicological and pharmacological profiles. So here we have two possibilities of interaction of the compound and the enzyme. And our cheminformatic data, values of enthalpy and free energy of Gibbs, and some experimental calorimetric data suggest that this pathway is the most preferential mechanism of action because of the reversible bond covalent bond. So the concerted step is a preferential. So we can suggest this mechanism of action. And nowadays, we can find 19 drugs with nitrile as well had in drug bank. So we believe that we stay in the right pathway. Okay, what we say today it's we have assessed the biological profile for the dipeptidyl trial derivatives with selective activity against the parasite in cell-based assays. So these four compounds here. Note that we have changed in the structure of our prototype, especially in P3 position, and a replacement of carbonyl group for a CF3 group, as we can see here. So these two compounds, it's a modify of the odonacatib structure. Odonacatib, it's a cathepsin K inhibitor and is the dipeptidyl nitrile derivative that advanced to the very last stage in the drug development pipeline, reaching the clinical phase 3. So we change the structure and to see what happens. Okay, we performed several analyses with the infectious morphology. So we incubate the compounds and the cells, and we performed assays as cell viability, use MTT and flow cytometry, total proteolytic activity of cell lysates and the intact parasite cells, invasion assays, cytotoxic assays, and analysis of the stability of the compounds. So here we have the comparison of the inhibition of cysteine protease in lysate and intact parasite. We aim to characterize the selectivity and potency of the compound set. And we observed that all compounds were in activity at 10 micromolar using the cell lysate of the amastigote and trypomastigote cells, these two different morphologies. By contrast, the same concentration was used to study the proteolytic activity in intact cells, and we observed several differences. And this difference in potency, maybe it's related to the setup and the sensitivity of these two types of assays, especially because when we observe the compound reference, we observe the completely absence of signals in the fluorimetric assays. So, and these two compounds are irreversible inhibitors. So it suggests that our compounds form a reversible covalent bond. So besides the our fluorogenic substrate is not specific to cruzipain, this enzyme is the major cysteine protease in Cruzi, as I said. So the difference of level expression between the tripomastigote and epimastigote forms could explain the distinctive bioactivity of these two compounds. It still not explain the profile observed for the other compounds. So, for example, this is a weak inhibitor for both parasite forms. Uh, so it's probably it's would necessary to quantify the activity cysteine protease present in each morphology to confirm the hypothesis. So at 100 micromolar, these two compounds show selectivity inhibitions of the cysteine proteases from trypomastigote lysate forms. And dipeptidyl trials inhibited the activity of cysteine protease inside attacked amastigotes in time-dependent way. This time-dependent behavior indicates exactly that reversible mechanism of inhibition that I talk. And the alteration of fluorescence intensity inside the amastigote could be related to the stability of the parental compound. In theory, a higher amount of the parental compound would be available at the beginning of the experiment, and in a long time incubation, the concentration in the well would be lower if the compound has a low chemical stability or low biological stability, reducing the inhibitory effect and the fluorescence. So this hypothesis was verified using the qualitative analysis of the chemical stability for all compounds in the same conditions of the extracellular amastigote assays. So we performed this analysis using the HPLC system with a reversed phase as stationary phase. It's a C18 column, and we using the liquid-liquid extraction as the method of sample preparation. And we observed that for the compound 662 and 663, this hypothesis is not valid because after 24 hours, the area under the curve of these two compounds showed not variation. For these two compounds, is not conclusive, probably because of the matrix effect observed in the experiments. So we have look careful with these two compounds and look for other other method development and perform other analysis for these two compounds. So about the cytotoxic and selectivity, all compounds except NEX663 show significant selectivity to the triple-mastigote morphology compared to the host cell, as we can see here. The white bar is the host cell, and the gray bar is the triple-mastigote. So next 663, despite to inactivity, is a good inhibitor of proteolysis. If we look at this compound, we observe the opposite. It's a weak inhibitor, but it's cytotoxic. So maybe the correlation between the proteolysis activity and cell death is elusive. And also, just our reference compound, E64, had potence higher than benzonidazole in the viability study according our statistical analysis. And look of this table, the situation is that triclosan lineages present a wide range of virulence effects in humans and sensitive of drugs. So because of this genetic diversity, the biological activity usually determinate in different triclosan strains. So we performed the concentration response assays using the epimastigote of Tulahuan and Y strains. And in general, the Tulahuan strain was more sensitive for all compounds except NAC 666. And NAC 662 uh, had the highest selective index for amastigotes of Tulahuan and Y strains. And the value is also higher than benzonidazole value for Tulahuan strain, and it's equivalent to the Y strain. So the only difference between these two compounds is the stereochemistry in the CF3 group. And according to our stability assays, both are stable after 24 hours. So the difference in potence is related to only to the stereochemistry of the CF3 group. And we performed the concentration dependent response in three different situations. The host cell with 2 hours pre-treatment with the inhibitors in the white bar. The trypomastigotes with 2 hours of pre-treatment with the inhibitors, the gray bar. And the parasite infection in the presence of the compounds, the gray dark bar. And the most of assays when we take into account the mean of the inhibitions, the best result was achieved when the host cell was pre-incubated with the next 663 for 2 hours. And here and the next the 594 and the 662 were less potent than 663 when the host cell was previously incubated with the substance. Also, NEX 666 was the most potent substance when the trypomastigotes were pre-incubated with the compound. And again, these two were the most potent ones when added during the host cell infection. And if we compare our results here, we can see the dipeptide unit trial were non-cytotoxic. Therefore, the antiparasitic effect was selective to block the infection of Cruzi, especially for the next 663. So we can conclude that the dipeptide unit trial can inhibit the proteolytic activity of cysteine proteases inside the amastigote cell, but no correlation with the cell cytotoxic was observed. And the activity of cysteine proteases can be reduced for the triple mastigote cell lysate, but all compounds were inactive in the same experiment used the epimastigote cell lysate. And this class of compounds show itself more potent in the triple mastigotes than to do the epimastigote forms. And it's common triple mastigote forms showed lower total proteolytic expression. And these results reinforce the lack of correlation between the inhibition of the cysteine protease and cell death, suggesting that another mechanism of action could be occurring. And also, the parasite infection was suppressed by our compounds. in all treatments, showing that these compounds are reducing the infection rate in vitro. And also, we measure the intrinsic reactivity of our compounds, and it's a simplified alcohol system using the inhibitor and some thiol, cysteine or glutathione. And we note that nitrile as warhead show moderate reactivity, especially when we compare it with the other warheads as vinyl sulfone and aldehydes. And the direct correlation between this assay and the enzyme inhibition is not expected because we don't have enzyme here. But this high intrinsic reactivity may help us to explain the lack of correlation between the enzyme assays and the cell assays. So we need perform future studies to determine the in vivo activity profile of this compound, especially NEX 662, once they have shown to be promising candidates for the further development. So I would to thank you for attention. Thank you for the our group, the founding agents, and the organizing committee for the opportunity to present this work. So thank you so much.